The weird history of the cell. There are three parts of the cell theory. 1. All organisms are composed of one or more cells. 2. The cell is the basic unit of structure and organization in organisms. And 3. All cells come from pre-existing cells. It all started at the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at that time. One such person was a fellow Dutchman by the name of Anton van Leeuwenhoek, who heard about this microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens. Once Leeuwenhoek had his microscope ready, he went to town, looking at anything and everything he could with them, including the gunk on his seat. Yes, you heard it right. He actually discovered bacteria by looking at dental scrapings. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules because they looked like little animals to him. While Leeuwenhoek is staring at his slit gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. Hooke was a guy who really loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus, it is Hooke who we can take for the term cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we know now as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made up of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who studied sleight of animal cells under the microscope. After studying animal cells for a while, he too came to the conclusion that all animals are made up of cells. Immediately, he reached out through sending mail to other scientists working in the same field as Schleiden, who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them. As for the last part of the cell theory, the cells come from pre-existing cells, Schleiden didn't exactly agree to that thought, as he swore that cells come from free cell formation, where they just kind of spontaneously crystallize into existence. That's when another scientist named Rudolf Virchow stepped in with research showing that cells did come from other cells. Research that was actually, what are the right words to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak. Introduction about cells. Cells have their own little world inside them. They carry genetic information, they can divide. Many have functions and processes that their organelles, structures inside them, can take care of. On our planet, we can divide cells into two major groups. A cell can either be a prokaryote or a eukaryote. Bacteria and archaea are prokaryotes, but everything else, plants, animals, fungi, and protists, are eukaryotes. Both prokaryotes and eukaryotes have genetic material. Both have cytoplasm. Both have ribosomes, which are small organelles that make proteins. Both have cell membrane, which controls what goes in and out of the cell. But what makes them different is a big deal. Prokaryotes have no nucleus, which holds the genetic material and controls the cell's activities. Prokaryotes have no membrane-bound organelles. Membrane-bound organelles are fancy organelles like the nucleus, mitochondria, and the Golgi apparatus. Eukaryotes, on the other hand, do have membrane-bound organelles. a tour inside a cell. So we want to take you on a tour of the ride of your life into the inside of the cell. Now, to start our trip, we're first going to have through the cell membrane, also called the plasma membrane. It is semi-permeable, which means it only allows certain materials and molecules in and out. By doing so, it keeps things inside a cell stable, also known as keeping homeostasis. Now, inside a cell, we find ourselves in this jelly-like material called cytoplasm. It surrounds all of these internal cell structures, and you'll find it inside both prokaryotes and eukaryotes. Organelles that are just floating around the cytoplasm can have more support than you might think. Cells contain cytoskeleton, which is a collection of fibers that will provide support for the cell and its organelles. The cytoskeleton can even play a major role in movement. Moving through this cytoplasm, let's start with ribosomes. They are not membrane-bound organelles, and they are going to be in both prokaryotes and eukaryotes. And they make protein, which is really important because that's so much of genetic material. DNA codes for protein. Ribosomes can be free in the cytoplasm, but they can be attached to another organelle too, which we will talk about a bit later. We are now going to focus on eukaryote organelles, which means organelles that are membrane-bound. So this takes our travel to the big boss, the nucleus. In eukaryotes, it holds the genetic material as in DNA, for example. All cells have DNA, but if a cell is a eukaryote, then there is a nucleus to put it in. The nucleus controls the cell activities. Inside it, it has nucleolus, which is where ribosomes can be produced. Attached to the membrane of the nucleus, or the nuclear membrane, you can find the endoplasmic reticulum, ER for short. It does a lot of processing of molecules for the cell, like protein folding, and it also is highly involved in actually transporting those molecules around, like a highway. There is rough ER, which has ribosomes attached to it, making it, as you can imagine, rough, and then smooth ER, which doesn't have ribosomes. Rough ER specifically tends to be involved with protein producing and transporting, because remember that ribosomes make protein. Molecules that leave ER can be sent away in vesicles that actually pinch off the ER themselves. Smooth ER has many additional roles, including detoxification, which is one reason why your liver cells tend to have a lot of smooth ER. Another additional role of smooth ER is that it can make some types of lipids. Next is the Golgi apparatus. It is the ultimate packaging center. It can receive items from transferred vesicle that pinch off the ER. It has enzymes that can modify molecules it may receive and sorts the materials it receives as well. It can determine where to send those molecules, including some that may eventually be sent to the membrane so they can be secreted, which means items that can be sent out of the cell. So with all that's going on in here, you might be wondering what's powering this thing, the mighty mitochondria, or mitochondrion if talking only about one. They are like power plants. They make ATP energy in a process called cellular respiration. It's not a type of power plant that you would think of. It runs on a glucose, which is a sugar, and needs the presence of oxygen to efficiently make ATP energy. Now at this point, we need to mention that eukaryotes are not one size that fits all. Animal cells can have differences from plant cells. We have a fork in the road here. For example, plant cells don't only have mitochondria, but they can also have these awesome organelles called chloroplasts. Chloroplasts actually make glucose by using light energy in a process known as photosynthesis. They tend to have a green look to them because they have a pigment that captures light energy and reflects green light. Both plant and animal cells can have vacuoles. Now vacuoles can have a lot of different functions, but many types act as storage of materials. Plant cells have one large vacuole called a central vacuole, while animal cells can have several smaller vacuoles. Remember how we already said that all cells have membranes? But plant cells additionally have a cell wall, which is a layer that offers additional protection and shape maintenance that animal cells do not have. How do we get out of this animal cell we have been in? Well, we could get out like a protein would. So if we were a protein, we would only be made because of the instructions from DNA. And remember that in eukaryotes, DNA is found in the nucleus. We would be made by a ribosome. The ribosomes would be attached to the rough ER, and then the rough ER highway would provide a vesicle to send us to the Golgi apparatus where the sorting takes place. And if we're tagged for being secreted, we're sent off through vesicle from the Golgi to the membrane. Out we go!